Ms. Patterson here. Today we're going to go over active transport, AP Biology Unit 2. So here I have a plasma membrane, and if you remember this plasma membrane, I have my hydrophilic heads, my hydrophobic tails, and material can pass through this membrane. I have some that can go with the concentration gradient. I have a lot of molecules here. I have a little bit of molecules over here, and it's going to go with its concentration gradient. So simple diffusion, I'm going to have material go through. I can have facilitated diffusion. Remember, these are proteins that are going to allow material to go through. And these are all passive transport. These are spontaneous. This is going to happen naturally. I have a lot of molecules and I have less molecules or solute. I have a lot of solute, less solute, and I'm naturally going to go through spontaneously. I don't need any help. But here, sometimes I have to transport molecules against its concentration gradient. And if I go against its concentration gradient, I'm going from an area where I have low number of solutes to an area where I have a high number of solutes. And sometimes I need that for certain processes in cells. So we have to do what's called active transport. And that's going to require the use of ATP or some type of electrochemical gradient that's going to allow material to come through. So let's talk about some of this. So this is going to allow material to where I can go from a high to a low. I'm going to keep pumping material into an area where I have a lot, I have less over here. Because remember, solutes, they want to go this way, right? They want to go over here where I have less. But this ATP is going to force these solutes to the other side, and I have to have energy to do that. I can use pumps, I can do cotransport, exocytosis, endocytosis. These all require energy and are a form of active transport. Diffusion, osmosis, those are spontaneous. They're going to go from a high to a low, but active goes from a low to a high. Here I have adenosine triphosphate. This is ATP. And this is the currency that is needed for a cell to do energy. I need this, like, think of ATP like a coin. If I go to a machine or a game, I got to put a coin in to get it to work. And so this is kind of like a coin for the cell. I put this in and work gets done. I'm going to be able to produce some type of energy to be able to allow something to happen. ATP causes what's called a conformational change to a transport protein. So if you remember proteins, long amino acid chains, they fold in a very specific 3D shape. If I change the shape of a protein, I change the function. And what happens is one of these phosphates get popped off, and this is going to attach to my transport protein. If I add this phosphate, I change the shape of the protein. And if I change the shape of the protein, I can change kind of the way it works. I do a conformational change. Later, this will leave the protein, this will come off, and then it will go back, it will conform back to the way it was. So this is enough to give it just a little bit of energy to allow the cell to do work. So there's different types of pumps. We have membrane potential, and this means that I'm going to have a different in electric charges across a membrane. So look at all of these H pluses. You remember what those are? Those are just hydrogen protons. I have had my electrons ripped off, and so these are just a positive proton floating in this solution. So this has a charge. I have some over here too, and I have a charge. These differences in charge can also help produce a pump, and I can change membrane potential. We call this an electrochemical gradient, and I can create a chemical force. The ion's concentration gradient. Remember, these are ions. An ion is an atom where I have a different number of electrons than normal. So these don't have electrons anymore, so that's not the way hydrogen is on its own. So this becomes an ion of hydrogen, or a hydrogen ion. This will also create a voltage gradient where it attracts positive ions and repels negative. So you see these negatives right here? This is creating a negative charge. And remember, positives love negatives. So these positive hydrogen protons are going to be attracted to this area. And look what happens to be here, this little proton pump. So ATP is going to come along and it's going to help push and shove these hydrogen protons through. Because remember, they don't want to be over here. There's a ton of stuff over here. These all want to come over here where there's more room to spread out. But this proton pump is going to keep pumping all these pieces into it. And these hydrogen protons are going to be attracted to this negative side of here, and then it's going to also repel negatively charged. So I'm going to be able to get more positively charged material by this proton pump. We're going to see this a lot in photosynthesis and cellular respiration. We see this type of electrochemical gradient. Another type of pump with electrogenetic pumps are going to cause that voltage gradient. It's going to attract positively charged ions, so these are going to get attracted, so they're going to be close to this pump. ATP is going to allow it to push through. So inside the cell is negatively charged. So here's the inside of my cell. I am negatively charged. The positive ions on the outside, these positive ions here are attracted also to this negative charge here. So they're going to stay here around this membrane, which is kind of nice because later on when we talk about ATP synthase, we talk about these hydrogen protons need to kind of stay around here for ATP synthase to work. So this charge on this membrane allows these hydrogen protons to come closer. We have another type of pump. We have what's called a sodium-potassium pump. These are a pretty common pump inside a cell. And we have an area where we have to pump from an area of low concentration to high concentration. I want a lot of these sodium ions, these sodium cations. Remember, cations are positively charged ions. I want a lot of sodium cations out here. Over here on the other side, I want a lot of potassium cations. Okay, potassium charged, positively charged ions over here. They don't naturally want to go. So I have to force them to go in that direction. So I have a protein that is going to be right here, and it's going to open up. And it's going to be charged where it's going to allow these sodium particles, these pieces, cations to come in. ATP is going to come along, and one of those little phosphates is going to attach to that protein. I'm going to change my shape. I change my function. So it's going to cause a conformational change. The bottom's going to seal up, the top is going to open, and then these sodium cations are going to get released outside the cell. When it's open, see that phosphate's still there. When it's open, these potassium cations are going to come in. And then when they come in, this phosphate's going to leave. And when it leaves, now I no longer have that phosphate there. I'm going to have another conformational change. This part here is going to close up. This part here is going to open back up. See, that phosphate's gone. And now these potassium cations are going to come into this solution inside the cell. And then it will work again. As it's open up, these sodium cations will come in. ATP will touch it. It's going to conform the shape. And the process is just going to keep happening. So it's going to pump sodium cations out of the cell, potassium cations into the cell, and we use this for nerve transmissions. So here I get 3 sodium cations, see 1, 2, 3, are going to get pumped out of the cell. I'm going to get 2 potassium cations, see 1, 2, are going to get pumped into the cell. And when I do that, I get a net charge. So I'm going to have more of a charge out here because see, I got rid of 3 out here and I only brought in 2. So this is going to create a charge on my membrane, which is going to help drive the pump. Another type of pump is what's called a protein pump. Again, remember, these are all just protons. It's just hydrogen ions. I have no electrons, so these are just one proton in this solution. So the protons are going to build up a proton gradient. And so it's going to pump hydrogen protons out of the cell. So ATP is going to come along and it's going to pull these hydrogen protons through this pump. These do not want to be here. Remember, they want to come this way, but this pump isn't letting it. It's going to take these and it's going to pump them through. These protons are going to be attracted to this negative charge. These protons are going to be also attracted to this negative charge, so they're going to stay around this membrane. So we see this in plants, fungi, and bacteria. And we see this with a product called ATP synthase. So you can see right here, this is pumping all of these hydrogen protons out into this intermembrane space. So we see this in mitochondria, we see this with plants, animals. We see this a lot, this proton gradient and this ATP synthase. I need to make ATP. So these hydrogen protons are going to come in. They're just filling up this area. They don't want to be here. And look, all of a sudden, ATP synthase is a protein that's being like, hey, we can let you come on through. Go ahead and come on through. So these hydrogen protons are going to start flying through this ATP synthase and cause it to spin. And as it causes to spin, it's going to use this energy of this flow of hydrogen protons to attach ADP. He's going to take one of those phosphates and make ATP. We're not here yet. Don't worry about that. But I'm just showing you, like, this is a good example of when we use a hydrogen proton pump. And we're going to talk about a lot when we start getting into cellular respiration and photosynthesis. All right, cotransport. This is going to be one solute is moved through ATP or electrochemical gradients against its concentration gradient, but another solute tags along. So let's look here. I have my proton pump. Okay, remember I have my hydrogen protons here. I have a few. I have a lot of hydrogen protons over here. This does not want to go this way. So these protons are going to be forced. ATP is going to come along and it's going to force this piece through. I'm going to have a huge concentration of hydrogen protons over here and they want to go back. And then look, ooh, I got a cotransporter here. So now what happens, these hydrogens are going to want to come back this way. So they're going to go with their concentration gradient in this direction. But now, all of a sudden, I've got sucrose. Sucrose is going to get tagged onto one of these hydrogen protons, and it's also going to get pulled through this cotransporter. So now I have lower concentration of sucrose here. I have a higher concentration of sucrose here. I can move sugar through this transporter by the natural flow of hydrogen protons. It's going to diffuse through this. So this is going to be spontaneous for the hydrogen protons, but as it spontaneously goes through, these sugars are going to piggyback on it, and it's going to force more sucrose into this area where it does not really want to go. So we're going to see this a lot with plants. Hydrogen pumps are going to bring sucrose. It's going to use the favorable movement of this diffusion. It's going to be a spontaneous of these hydrogen protons to come through. And it's going to be moving this part here where it doesn't want to go, but it's going to force it through. I can transport large molecules, exocytosis and endocytosis. So exo means out. I'm going to have this little vesicle. Remember, this is a phospholipid, a plasma membrane. It's going to come and it's going to fuse to this membrane. Remember, they're like soap bubbles. And this soap bubble is going to fuse to this big soap bubble. And it's all going to become one membrane. When it does, it's going to open up and the solutes, the material inside, is going to be put out into the cell. Endocytosis, I can bring stuff in. Stuff's going to come to the plasma membrane. It's going to fold in. You're going to have like these little pseudopod feet are going to come in and they're going to pinch off, and then it's going to bring it into the cell. We see this with bulk transport and polysaccharides, which are really big sugars. proteins, bacteria. We're going to see this with exocytosis, endocytosis. Big material has to go through some type of transport, and this needs energy to be able to do that. So let's look at exocytosis. I have a vesicle from inside the cell. It's going to fuse with that plasma membrane, and it's going to push or expel its contents outside of the cell. So here I have my vesicle, I have my material in here. This happens to be a neurotransmitter, and this is going to fuse to the membrane, and then it's going to push that material, that neurotransmitting signal across this membrane, and it's going to allow it to leave the cell. Vesicles are going to fuse to this membrane, and then this vesicle, again, see how this was one circle, and now you see how the green is now a part of this. This phospholipid bilayer is now going to be a part of that membrane. And the substance will be released out of the cell. And we see this a lot with nerves, neurotransmitters, jump in this synaptic gap. So here I have a nerve. These are going to come to the end. They're going to fuse, and as they fuse, they're going to push their material across. This will get received by the other side of this neuron, and then I'm going to have information get passed. So I'm going to have these vesicles will fuse and become a part of that membrane. Endocytosis, endo means in. So endocytosis, I'm bringing material into the cell. So I'm going to be forming new vesicles from plasma membrane. So I have the plasma membrane, material is going to come in, the membrane is going to fold and it's going to pinch off, and then I'm going to have this plasma membrane that was part of the cellular membrane is now a vesicle or a vacuole. It just depends what it's going to be used for, and now it's a part of that. I can take substances, usually macromolecules, they're going to be brought into the cell, and that's going to be when the vesicle fuses with the plasma membrane. So we see this with phagocytosis, pinocytosis, and receptor mediated. So phagocytosis is when I get food, pino is going to be liquid, and receptors is when I'm only going to be very selective in what I can bring in. So phagocytosis means cellular eating, and that's when a cell engulfs a solid particle, that's your key word right there, into a vacuole to be later broken down by lysosomes. So what happens is this will come in, the plasma membrane is going to pinch off, this is going to go into the cell. Lysosomes, remember those are the organelles that break down and recycle, are going to fuse to this vacuole. And now it's going to become one big bubble. And those lysosome enzymes are going to start to hydrolyze or break down this food source to be used later on. And wastes that are going to be created will be later on expelled out of the cell. Another example is the amoeba. It has these pseudopods. They'll go in, and these pseudopods will engulf and go around, and then it can start to break down food. So here we have an amoeba. I have bacterium. I'm going to produce what are called pseudopods. Pseudopods, these little fake feet. They're going to go around and engulf, and I'm going to produce a food vacuole. And then I'm going to have lysosomes will come in, they're going to fuse, and they're going to start to break down the material and recycle what it can use, and whatever it can't will later on be released out of the cell. So with phagocytosis, here's an example of me having food vacuoles and lysosomes coming in to break down. You can see that I have my food vacuole, lysosome is going to come in, it's going to fuse, it's going to become all one piece, and then whatever gets recycled will get reused, and then whatever isn't is pushed out of the cell. So they're going to use vesicles to package food. And then this is a good example again of compartmentalization. This is allowing lysosome to come in, to fuse, to break down what's inside this vesicle, so it's not interfering or stopping other processes from happening inside of the cell. Here I have pinocytosis, which means cellular drinking, and this is going to bring in these dissolved substances or solutes from other fluids outside the cell. So this is outside the cell, and this is inside, and you can see all this water, and I have these solutes inside, and these solutes can get brought in. Now, it's not as selective, so I could get, if I want triangles, I might get a few stars mixed in with it too. But I'll try to get as many of these little squares as I can. So this is going to put them in a protein-coated vesicle, which means it's going to be a little bit thicker. It's going to prevent stuff from transferring through that plasma membrane. All right, and the protein coat is going to help transport the molecules. So it keeps this stuff inside of this vesicle and doesn't allow it to pass through as easily. We have receptor-mediated endocytosis. And here you can see on this plasma membrane, I have these little hooks. And these hooks are going to hook a very specific type of what's called a ligand, or a type of substrate, molecule, solute. Something's going to come in that the cell wants. And it's going to be very selective. So here I'm going to come in, I'm going to grab these stars. If one of these squares hit it, it's not going to collect, it's not going to take it. So these will only collect stars. And when I have them, now I'm going to have them close in, and this vesicle is going to pinch off, and all I'll have inside are stars. So here you can see these little receptors. They're going to come in, they're going to have a very specific solute, very specific molecule. It's going to fold when I have enough, and it's going to pinch off and go. And here you can actually see one kind of doing that with an electron microscope. So these are going to be receptors on the plasma membrane. They're going to bind to very specific solutes or molecules, and they're going to allow more of one type of product to be brought into the cell. They're also going to be coated, so these will also be coated to keep them from being absorbed or passing through that membrane, and I can bring in a large amount of these substances into the cell. All right, that's it for this one. So active transport. If you guys have any questions, let me know. We'll talk to you later. Bye.